Hello everyone, my name is Nisrin and I am a master student at Biotechnology Institute, Ankara University. Today's talk is entitled Salmonella Biofilm Dispersal, an approach for biofilm control. First, I will start with an introduction about Salmonella and biofilm life cycle, followed by how dispersion is used as biofilm control strategy, and then I will talk about the genetic regulation of Salmonella dispersal and the methods used currently to disperse Salmonella biofilms. I will finally propose recommendation for further studies. Salmonella enterica is one of the most important foodborne pathogens leading to outbreaks worldwide. It forms robust biofilms on both biotic and abiotic surfaces in the medical and industrial settings. Biofilms are cell populations attached to a surface and encased in a protective self-produced polymeric matrix. This matrix protects the cells residing in the biofilm from high concentrations of antibiotics and harsh environmental conditions. As a result, biofilm-forming pathogens such as Salmonella are very resistant to conventional treatment methods, making the biofilm-related Salmonella infections a significant challenge in healthcare. In order to control this structure, we need first to understand the biofilm life cycle. The biofilm development starts with an initial attachment followed by irreversible attachment, where cells start to multiply and produce the polymeric matrix. Then the biofilm maturation occurred and a mature, robust biofilm is formed. Eventually, bacteria residing in the biofilm undergo dispersal, in which again planktonic cells are released into the environment. Biofilm can be controlled following two avenues. The first one is the inhibition of biofilm formation, in which the irreversible attachment is inhibited by several strategies. One of them is surface coating. The second one is the removal of existing biofilms. The eradication of the already existing biofilms is hard because it requires higher antibiotic and antimicrobial concentration and leads to higher costs. That's why the biofilm eradication strategies needs to be enhanced. So now, the dispersal, how can the biofilm dispersion be used as a biofilm control strategy? First, biofilm embedded cells undergo dispersion either passively by mechanical action or actively as a response to external or internal signals. While dispersion, the biofilm releases more vulnerable cells to the environment. The dispersed cells are more sensitive to antibiotics and antimicrobials comparing to biofilm cells or planktonic cells. Thus, inducing the biofilm dispersion is actually releasing vulnerable cells to environments that can be treated easily by conventional methods. And that's why it is considered a promising strategy for biofilm eradication. Biofilm dispersal strategies usually target the extracellular polymeric matrix, the quorum sensing system, or the dormant cells. Targeting the extracellular polymeric substance matrix is actually targeting the component of this matrix, which are proteins, DNA, and polysaccharides. Targeting these components is achieved by enzymes. These enzymes can be DNases, proteases, or glucoside hydrolases. Targeting the quorum sensing system, which is a system of communication between bacteria using chemical signals called autoinducers, is common too. This system can be targeted by inhibiting the synthesis of these chemical signals or degrading or modifying these signals, inhibiting the bacteria from communicate with each other, and thus inhibiting vital processes such as biofilm formation and virulence. Third, targeting the dormant cells. The dormant cells or the persister cells are cells that are metabolically inactive. These cells are more resistant to antibiotics, and that's why the identification of antibiofilm peptides is very vital to target these dormant cells. Finally, the dispersal molecules are molecules that can induce a dispersal response inside a mature biofilm. One of these dispersal molecules is the nitric oxide donors, the cis-2-decanoic acid, the CDA, and the chelating agents such as IRTA. For an efficient removal of the dispersed cells, released during the dispersal and the remaining biofilm, dispersal agents are combined with antibiotics. These antibiotics aim to eradicate the dispersed vulnerable cells. The genetic regulation of Salmonella dispersal is not well studied. However, as many biofilm-forming bacteria, low levels of the bacterial second messenger cdGMP lead to dispersion. This cdGMP is synthesized by Dig1 light cyclases, and its high levels leads to biofilm formation. However, it is degraded by phosphodiesterases, and its low levels lead to dispersion. It was found that in Salmonella typhimurium, the levels of cdGMP is regulated by the carbon storage regulator A, CSRA. CSRA regulates the degradation of cdGMP signal molecule by positively affecting the genetic expression of phosphodiesterases. In the current literature, dispersion strategies of Salmonella biofilms are as follows. Nitric oxide donors and their combination with disinfectants, and CDA with its combination with antimicrobials are widely used to efficiently disperse Salmonella biofilms. Antibiofilm peptide that can block PPGPP signaling was identified to be an efficient strategy to induce Salmonella biofilm dispersal. Also, antibiofilm peptides such as nisin and their combinations with chelating agents such as ETA was also effective in dispersing Salmonella biofilms. Finally, a novel study identified an antibiofilm exopolysaccharides from Lactobacillus casei Ni2 strain that has the ability to induce the dispersal event in Salmonella biofilms. As a conclusion, we would like to recommend some research points for further studies. There is a need to elucidate the genetic and metabolic pathways behind the dispersal events of Salmonella enterica, because understanding the genetic and metabolic pathways will lead to better understanding how the dispersal agents work and better targeting of the dispersion signals. Second, in vitro and in vivo investigation of dispersal agents' efficacy on multi-species biofilms is crucial, because most of the research done in the current literature are on monospecies biofilms. But we all know that in clinical and industrial settings, most of the biofilms are multi-species, so further investigations is in need. Lastly, an in vivo determination of the safe concentrations of dispersal agents and the effectiveness of their combinations with antibiotics is vital in order to use and apply these dispersal agents in clinics against biofilm-related infections. Thank you very much for listening. If you have any further questions, don't hesitate.